Okay. Oops. Not good. Yeah.
All right. So that should be it.
Hey, doctor. You made it?
I knew no matter how late I would be,
Yeah.
I'd be the first one in.
Yeah, I know.
Sometimes they prop open the door, so I may go and
Oh, yeah, that's a good idea.
sort of let people in.
Do you mind actually propping the door for a couple minutes?
Well,
My wife is going to be here, and she's barely even going to know how to get to the building. And I have some homeless bums I want to show up. I'm expecting. Taking off? You saw the title, and you're like, I'm going to leave. Yeah, I know. That's an eternity with this stuff, yeah. Just have to make sure my committee shows up.
sorry, not suicidal yet?
There he is.
You're not ready for grad.
How you doing?
Projector's working, so I'm good.
6 months
Yeah?
of despair before
Alright.
Except it keeps working.
Yeah.
But then it's still hard. If it's a GMS student or would say like a dental student, probably dental.
Are we ready, or do we wait till the 5 after Bill? Okay. So we've arrived at this day with the help of a lot of people and innovation and support. And I want to just thank up front Barbara Schreiber, Linda Hyman, David Harris, and the two committee chair. We didn't notify Bob Nugent, but I'm sure he wouldn't be able to get down. And Matt Nugent, and Bob is here. And I'm happy to get to this point also to introduce again Anthony J. I was fortunate to have him join my lab when we were finishing a very complicated project that involved a lot of people over a long time, including Nasi Wong, who's here, and Sue Zhu, who finished her thesis. And there are just quite a few things that we couldn't wrap up, and we needed duplication and proof of principle. And Anthony came in, wrapped up a very nice paper, and it was sent to Biochemistry. It was the fastest paper ever accepted. It asked for revisions. I sent the revisions back 3 days later. It was accepted 12 hours after I sent the revisions. And then Anthony went on to develop really new areas that I hadn't put in my plan for him based on his experience that developed into the second major study. And that paper went into JBC with virtually no criticisms. And the editor contacted me 4 months later, the handling editor, and said that was the most cited paper, downloaded paper from the JBC website of all the papers I handled in the last 4 months. So, and he really did create a new and innovative area, and he went on to do other issues. He's not going to talk about everything he did today, but summarize those two areas and maybe some future directions. So, Anthony?
Good. According to the CDC, obesity and heart disease rates are rising. Here's the obesity heat maps. You can see 1990, 2000, 2010. So approximately every 10 years, it almost goes up about 10%. And mirroring the obesity heat map, you can see the heart disease death rate map, recent map, is similar. It's also been rising. And the predominant cause of heart disease death is arterial plaque development. Now this picture is from a researcher named Dan Stein, many of you know. It's a cartoon of plaque progression, the stages of plaque progression. So you can see it starts with lesion initiation, fatty streaks, fibrous plaques, and thrombosis. And patients here with these plaques also have been found to have elevated fatty acids. And this is just an example from our group of rabbit plaque progression. you'll note that oxidized LDL is present on each stage here. We'll talk about that. And then CD36 is expressed in all the involved cell types. So it's expressed in macrophages, myocytes, and endothelial cells. Here's the expression data for CD36. You can see again we got adipocytes, myocytes, macrophages, and muscle cells. So CD36 is a scavenger receptor and has a significant role in oxidized LDL binding and fatty acid metabolic fate. The oxidized LDL binding is well characterized and well established. All of these scavenger receptors in this class share the defining feature of being able to bind modified forms of LDL. And CD36 specifically, shown here, is intimately involved in lipid metabolism and homeostasis, strongly implicated in pathological conditions associated with metabolic dysregulation, including obesity, insulin resistance, diabetes, etc. So the rationale for this study is that CD36 is centrally involved in these prevalent diseases, atherosclerosis, etc., all of which have high circulating plasma free fatty acids and ox-LDL. And the previous studies with CD36 and fatty acids have led to various associations without understanding the mechanisms. And there's also been a lack of studies with CD36 and fatty acids on structure-function effects. So my overarching hypothesis for this project was that free fatty acids exert multiple effects on the function of CD36, both normal and pathophysiological. And to address this overarching hypothesis, I'm going to present 3 parts. Number 1, CD36 involvement in fatty acid transport across the plasma membrane. 2, direct binding of CD36 to free fatty acids. We'll use SPR. We'll look at that. And then number 3, the effect of CD36 binding on free fatty acids. Okay, CD36 binding to free fatty acids on CD36 binding to oxidized LDL. So we'll start with this transport across the plasma membrane. Now, there's 2 major hypotheses that have been promoted for CD36 involvement with fatty acids, or I guess fatty acid uptake across the plasma membrane. And one of them has been published by Abramad's group, and it's shown in this cartoon, and this has been published in various places. Fatty acids need CD36 to enter cells. In other words, CD36 is a transporter protein. And she also argues, Abramad here, she also argues that CD36 only has one fatty acid binding site. And the other major hypothesis for fatty acid crossing the plasma membrane is that is simple diffusion. So with or without CD36, fatty acids enter the outer leaflet of the cell membrane. They flip-flop across, and they release a proton in the intracellular space. So that's what we're going to investigate. Now, previous fatty acid uptake experiments have added radioactive fatty acids with or without CD36 expression. And then they essentially do endpoint measurements, and they ask how much fatty acid remains in the cells. So they measure essentially triglyceride. And our novel approach has been to use pH-sensitive dyes trapped inside cells or vesicles and study the real-time rate of fatty acid uptake. And this is an example of BCECF, which is a dye that's pH-sensitive, and it's trapped inside vesicles. So we make vesicles with BCECF, run them through columns, and eliminate the free dye. And the advantages of number 2 is that, yes, we can use protein-free vesicles. We can use live cells with CD36 or without CD36. And then it's in real time again. So when I joined the lab, I verified some of the previous experiments using protein-free vesicles, using this dual fluorescent assay approach. So here's BCECF. But first we add FPE to the outer leaflet of vesicles or cells, and that's also a pH-sensitive dye, and it does not flip in the vesicle, so it stays on the outer leaflet. And it detects the fatty acids entering that outer leaflet, and you can see here some of the data with, again, protein-free vesicles. And when you add oleic acid, fatty acid, you see a drop within seconds. So the fatty acids, in other words, are entering the outer leaflet within seconds. And This is BCECF data. And BCECF, again, is trapped inside the vesicles. And when you add oleic acid here, again, it's a drop within seconds. So we see the fatty acid entering the intravesicular space within seconds. So for cells, we express CD36 in HeLa and 293 cells, neither of which endogenously express CD36. And we express empty vector and CD36. Here's the Western blots for these. And we've also knocked it down with siRNA. And we find that it's plasma membrane localized in both of these cell types when we express it. And we look at it under the confocal. And we've also previously shown that CD36 expression levels in 293 cells are comparable with 3T3 adipocyte cells. And for the biochemistry paper, I developed this flow cytometry validation of the function of CD36. So we know it's expressed, but does it function? It's on the outer surface of the cells. So we added di-I oxidized LDL, so fluorescent oxidized LDL, for 30 minutes in cells with or without CD36. Fixed them and permeabilized them, and then did flow cytometry. And you can see there's significantly more fluorescence with CD36 expression, indicating more uptake of oxidized LDL or binding. So in other words, the protein is functional. And so moving on to this real-time assay, fluorescence spectroscopy. You can see here I have FPE on this top panel. And again, that's the dye that detects fatty acids coming in to the outer leaflet. Again, within seconds, we see oleic acid. With BCECF trapped inside the cells, you can see the fatty acids again. They're entering the intracellular space within seconds, and that's dose responsive. So it enters rapidly, releases a proton, and HeLa cells. So these are all 293 cells here. I repeated the experiment in HeLa cells, again with and without CD36 expression, with FPE, and with BCECF, and found it again within seconds. The fatty acids are entering the intracellular space. And we published this in Biochemistry, along with data from Sue, who was in our lab previously, and she quantified fatty acid esterification products with radioactive oleic acid, and she found that the expression of CD36 significantly enhanced triglyceride formation within 1 hour using those assays. And we also used Bo-Dippy staining, which showed that with CD36 expression, you have enhanced triglyceride droplet formation within 24 hours, size and number. So the conclusion for that was CD36 enhances fatty acid uptake by increasing the rate of intracellular esterification, but not the transport across the plasma membrane. In previous investigations, they've used these fatty acid uptake inhibitors, and there's several of them. These are small molecule inhibitors. And of course, that suggestion is in conflict with our new findings. And kind of the gold standard of these inhibitors is SSO. It's the most notable and widely used. And the structure is here. It has a fatty acid tail, and it has the succinimidyl oleate group, so sulfo-succinimidyl oleate. And you can see there's potential for hydrolysis here. It's a highly reactive and unstable molecule. It reacts with lysines, and here's the reaction. And SSO, importantly, is said to be CD36 specific. So our ensuing hypothesis is that this small molecule inhibitor, this SSO, inhibits metabolic functions rather than disrupting the plasma membrane transport, and people are interpreting that as inhibiting the uptake. So again, it's the most widely studied inhibitor. It's presumed to be plasma membrane impermeable, but nobody's actually looked at that. And it has been found to interact with the extracellular domain of CD36, lysine. And our experiments here with SSO aim to distinguish whether it's entering the cells and whether it's modifying other proteins, which could potentially disrupt metabolism. So first, I did an experiment with SSO hydrolysis and found the rates are relatively slow. You can see the cell buffer and the SUV buffer, the vesicle buffer. It's certainly within minutes. And it is, again, unstable and reactive. but the hydrolysis occurs within minutes. However, using our pH assay for the measurement of molecules across the plasma membrane, SSO enters the intracellular space within seconds, so we also think SSO is flipping into the across the membrane with that long fatty acid tail. And we have other data from our lab on this, and previous lab members have found this also occurs in protein-free vesicles, and this is the data here. This is just oleic acid, and this is SSO in protein-free vesicles. You can see it's causing a pH drop. And this arrow points out nigericin, which is an ionophore, so we're punching holes in the membrane to release the protons back out, just to show that it's a true pH drop effect. And we've also done, our previous members in our group have also done this experiment with SSO-treated vesicles. And then they add fatty acid and find the fatty acid in those cases is still within seconds, still rapid. So to further investigate this hypothesis, SSO is actually entering cells. We created and applied a novel SSO antibody raised against a peptide cross-linked to SSO. So it's a peptide with 3 lysines. And we've done dot blots on this, I'm not going to show. But this is the microscopy, confocal microscopy. With permeabilization, here's a control. The permeabilization is to allow the anti-SSO antibody to get through the membrane. And you can see DAPI just lights up the nucleus to show that cells are here. With or without CD36 expression, with no SSO treatment, we get a little faint nuclear background binding, but essentially no binding of the antibody. But when you treat this live cells with SSO and then fix them and go through this experiment, you can see there's some dose-responsive binding. And again, this is even without CD36. So we're seeing binding just to other proteins on the surface of the cells. And then when you express CD36, I did this with and without permeabilization to kind of emphasize the CD36 on the surface and then other proteins that are intracellular. And again, I use different doses, and we find green, which indicates SSO binding, red, which indicates CD36, and then yellow, which is colocalization. So we get a lot of binding to a lot of different places, indicating, again, SSO-modified other proteins. So the major conclusions from this section is that CD36 enhances fatty acid uptake by increasing the rate of intracellular esterification, but not the transport. And then inhibitors of fatty acid uptake may be inhibiting intracellular metabolism rather than plasma membrane uptake, and this part of the investigation is ongoing. So the next section, we're going to look at direct binding of CD36 to free fatty acids. And the hypothesis here is that CD36 binds multiple fatty acids in the extracellular transmembrane domain. And this is a solution structure done by our group previously of fatty acid binding protein. And it has 2 oleic acids bound, and the reason I show this is because CD36 has a homology domain to this region of fatty acid binding protein. And again, there's 2 fatty acids bound here, and they bind with different affinities. Our approach for this is surface plasmon resonance. So surface plasmon resonance uses a prism to break the light, to bend the light, and you have proteins covalently linked, in our case CD36, covalently linked to the chip, and you flow analytes across the surface, and if that changes the mass-charge ratio, this light will shift, and you pick that up on the detector. You can do multiple analytes in different wells, and in our case with the BioRad machine. And the beauty of this is it gives you real-time association and then dissociation when you wash out the fatty acids. So the difficulty, of course, with fatty acids is solubility. So we've previously developed a novel delivery approach, which is methyl beta-cyclodextrin in aqueous PBS buffer. Members of our group previously have found that fatty acids are rapidly delivered to and extracted from membranes with cyclodextrin. And this is shown again with pyranine, which is another pH-sensitive dye. And we've studied various fatty acid types, and it's always rapid. The delivery is rapid. This is what cyclodextrin looks like. It looks like a donut that essentially the fatty acids insert into. And These are some of the fatty acids we used. You can see we got fully saturated, we've got mono unsaturated, we've got oxidized fatty acid, HOD, we've got trans fatty acid, aleotic acid, DHA, EPA, so we have omega-3 fatty acids, alpha-linolenic acid, and then we have omega-6 fatty acids, arachidonic and linoleic. And some of these, of course, have different solubility, so we use different levels of cyclodextrin. And our running buffer was 200 millimolar PBS to eliminate a lot of the background binding to the SPR chip. All right, so this is somewhat of a control experiment here, where in these wells we linked HSA, which is human serum albumin, to the chip, and then, of course, CD36 on this one. And we just simply flowed different doses of methyl beta cyclodextrin across the surface to see if cyclodextrin was binding our proteins, and we find it's not binding, all the way up to 5 millimolars of cyclodextrin. And these are subtracted to the empty well, which does not have any protein bound to it. And then when we add oleic acid, again, albumin, of course, binds. This is a positive control. And CD36 we find also binds the oleic acid. And then IgG, which is a negative control, essentially showed no binding. So oleic acid binds. So then we looked at some other fatty acid types, trans fat, HOD, palmitic acid, and DHA. And we found, of course, albumin binds all of these. And you can see there's some biphasic binding, which means there's initial rapid phase and then some slower binding that occurs. But it's interesting because HOD was the one that did not bind CD36 in a dose-responsive way, but all the other ones did. And we've looked at some of these other omega-3 and omega-6 fatty acids, and we find binding here. So the major conclusion from this section is that CD36 binds various structurally distinct fatty acid types, including trans fat, fatty acid, saturated, monounsaturated, polyunsaturated, but it does not bind oxidized HOD. And again, the dissociation of this binding was rapid, but the Ka and Kd can be considered only as estimates in these experiments because of the contribution of cyclodextrin and likely binding sites on CD36 and HSA. And based on, again, some of that biphasic binding, and then comparisons with albumin, which of course has 9 fatty acid binding sites, CD36 is likely to have multiple binding sites to fatty acid. So the third part is the effect of CD36 binding to free fatty acids on CD36 binding to oxidized LDL. The hypothesis here is that CD36 binding to the fatty acids leads to enhanced CD36 binding to oxidized LDL. And the rationale for that is that despite short intracellular domains on CD36, signals are still transmitted when extracellular ligands bind. And both fatty acids and oxidized LDL the binding domains are present in this extracellular domain. And they're both in the same region. The ox-LDL binding and the fatty acid binding portion of CD36 are in the same region of the protein. So again, using SPR, this time with oxidized LDL, flowing oxidized LDL across instead of fatty acid, IgG essentially shows no binding. CD36, of course, binds oxidized LDL. This has been known, but this is the first time anyone's shown it on SPR. And then I want to emphasize here that let's just take one peak. In this case, 4 nanomolar ox-LDL. It looks to be about 150. You can see that. So this is the same experiment, except I'm adding 150 micromolar of oleic acid. And again, with 4, you can see the oleic acid here. With 4 nanomolars of oxidized LDL plus the oleic acid, rather than at 150, now it bumps up to about 400, and that occurs with different doses of ox-LDL. So in other words, the binding is significantly enhanced when you add oleic acid, fatty acid. The oxidized LDL binding is enhanced far beyond what just pure oleic acid would give you. So moving on to cells here, we did confocal microscopy with cells expressing CD36. These are 293 cells. And oxidized LDL, shown in black and white here and then in color here. And we find with serum present, we have significant oxidized LDL binding. But if you have serum-free media and you add ox-LDL and incubate, you lose the binding. And you also don't have the binding when you have charcoal-stripped serum. And you add, again, fluorescent oxidized LDL. But when you have that same situation with serum-free media and you add back just oleic acid to that, you rescue some of the oxidized LDL binding, and it is dose responsive. And then, of course, when you don't express CD36, you lose the binding. And so we looked at three-dimensional confocal microscopy here to investigate, is it just binding, or is it actually being taken up? The oxidized LDL is being taken up by CD36. And we think the uptake it's actually oxidized LDL uptake. And so this is CD36, and in this case we use GFP tagged CD36. The antibody binds the same site as oxidized LDL to a degree, so rather than use antibodies, we just use GFP CD36, oxidized LDL, and you can see in our three-dimensional Z-stack, the CD36 is more at the surface, and we think the oxidized LDL is intracellular, so it's uptake. And then finally, we moved on to, again, trans fat, HOD, palmitic acid, DHA, so other fatty acid types. And you'll remember that CD36 did not bind HOD via SPR. But you can see here, it's interesting that it also does not increase the oxidized LDL uptake. And unexpectedly, also DHA did not increase the oxidized LDL uptake in this assay. So we've done some preliminary experiments with different omega-3 versus omega-6 fatty acid types, and these ones do all bind via SPR. And you can see with our omega-6 fatty acids, we get some oxidized LDL binding and uptake, and with omega-3, it's diminished. So we think that's an important aspect here. And then finally, we reduced the disulfide bonds, and they did not alter the binding of CD36 to oxidized LDL. So this is just a standard oxidized LDL binding assay. You have CD36 on a 96 well, and then you just add different doses of oxidized LDL. And again, with or without reduction, you still have dose-responsive binding of oxidized LDL. And the reason we did that is because the disulfide bonds are critically important for CD36 binding to malaria-infected red blood cells. So there is precedent for the disulfide bonds on CD36 to be important for binding. In this case, we believe that that's on the opposite face of CD36 on the structure. This is a CD36 model based on LIMP2 homology. And we think that this is our proposed mechanism for CD36 or for fatty acid and oxidized LDL collaborative binding, where fatty acid comes in and opens some kind of a face on CD36, and that allows oxidized LDL to bind. So the major conclusions from this section is with CD36, oxidized LDL binds CD36, and the oxidized LDL, that binding is strongly enhanced by addition of fatty acid. Serum enhances that binding, and again, it doesn't occur with charcoal strip serum. All fatty acid types investigated enhanced the dioxidized LDL binding to CD36 except HOD and omega-3 fatty acids. And then again, disulfide bond reduction did not alter it. So here's a summary table of the combined SPR binding and oxidized LDL microscopy results. We find essentially CD36 binds to all the fatty acids except for HOD. But these fatty acids don't enhance ox-LDL uptake in all the cases. It's HOD, but also, again, the omega-3s. So the major conclusions from the three parts combined here is that CD36 does not enhance the rate of fatty acid transport across the plasma membrane, but it does enhance the lipid droplet formation. And CD36 binds most structurally distinct types of free fatty acid except HOD. Binding of ox-LDL is enhanced by free fatty acids. HOD does not bind CD36, nor does HOD enhance that uptake. And then again, the omega-3 versus omega-6. So future directions, we should look at the metabolism. So using mRNA screening and likely cell signaling pathways, we need to investigate CD36 metabolism, especially that initiation of lipid droplet formation. And again, possible metabolic losses due to that uptake inhibitor, SSO. Using confocal microscopy and Q-dot nanoparticle tracking on CD36, we need to investigate the variation in plasma membrane localization, which also changes with malaria-infected red blood cells. And we want to look at that upon addition of different types of fatty acid, along with oxidized LDL, and look for possible localization changes. And then using SPR, we need to further investigate the binding of CD36 to oxidized LDL in the presence of various fatty acid types, especially with this omega-3 and omega-6. And then finally, we need to do structural studies of CD36 using hydrogen exchange mass spectroscopy. And structural studies have not been done on CD36, and it would contribute to the whole field because CD36 binds obviously fatty acids, but it also binds an oxidized LDL, but it binds things like malaria-infected red blood cells, it binds amyloid, beta, and a list of other things. And hydrogen exchange essentially works because the hydrogens along the protein backbone exchange, and so you use heavy water, deuterium-labeled water, and exchange out the hydrogens, and then you can look at the proteins and find which ones are exchanging and deduce essentially which ones are exposed to the surface, the water, and which ones are hidden. And John Engen at Northeastern has this interesting technique where he uses nanodiscs to insert plasma membrane proteins. So this would be an ideal study because it would give us native structure in an actual plasma membrane. So we're working to purify the protein for this. And to continue this work, an editorial was recently published in ATVB supporting our hypothesis of fatty acid diffusion. The work is supported an ADA grant submission and an R01 grant submission. And of course, I'd like to thank the committee members, past and present, which includes Matt Nugent, and my lab members past and present, and then the various students over the years. Thank you. Questions? Dr. Tornheim.
So if the fatty acid is binding to CD36, where the oxidized LDL also binds, and it's enhanced. So this implies that close in, they're not at exactly the same point.
Right.
And
Well, it may be just trapping the fatty acids in that barrel structure. I don't know. I mean, I'm just thinking of the fatty acid binding protein homology.
but the fatty acid, I was thinking not so much the trapping of the fatty acid, but didn't you say that the binding of the fatty acid enhances the binding of the oxidized LDL?
Right.
And so, any idea whether that's just a conformational change on the CD36, or is the fatty acid able to do a bridging of some sort?
It's a great question. I don't know, but it's something we should definitely look into.
Is it more than one fatty acid?
I think multiple fatty acids contributes to enhanced binding. Yeah. Yeah. Yes.
Very interesting. Maybe you could comment a little bit more. I think you started off with cardiovascular disease, and your model systems seem to be cells that lack CD36 for gain of function. So my understanding, at least from the knockout mice maybe 20 years ago, the primary phenotype was the inability to take up fatty acids. Right?
Yeah,
So
but the
think
mice also have increased cardiovascular problems.
Right.
Yeah.
So do you think there's a possibility that your cell system is missing another protein component that's important for the native CD36 to perform its function?
It could be. I mean, there is precedent because alkaline phosphatase in the intestine has been shown to interact with CD36 in terms of phosphorylation. And there's some binding, not with fatty acid that's been shown, but there are some other proteins that they've looked at. And that would be one way to look at that, I think, would be the phosphorylation. But with HEC293 cells and HeLa cells, they don't express other scavenger receptors, and that's one of kind of the reasons we chose those, is to hopefully eliminate some of those other, at least with the scavenger receptors. I know you're not asking about those specifically, but yeah, that's definitely an interesting area. And Barbara had her hand up.
Yeah, so you, the first part of your talk, you allude to metabolic implications of CD36, aside from what's been speculated before. And then in the last part of your talk, you said what you would do to look at messages, et cetera, but you must have some hypotheses as to what these metabolic changes might be that lead to this accumulation of triglycerides.
Right. So in some of our experiments, we do add the acyl-CoA synthetase inhibitor, triaxin C, and it doesn't affect the fatty acid uptake, of course, and it also doesn't affect the binding of oxidized LDL to CD36 with the enhanced binding with the fatty acids. But we do suspect that's involved somehow, the acyl-CoA synthetase. But, I mean, like I say, I think the approach would be to do a screen and to see what we get from that. Yeah. Haya?
Two questions. In your cellular studies where you tested, which is nice, you did the fluorescence studies using DII oxidized LDL, but the question is what happens with the LDL without the fluorescent probe, because you can measure obviously uptake by looking at accumulation of cholesterol esters. So
I haven't done that. It might be worth doing, especially as we move on to different fatty acid types and try and distinguish. In fact, I think it's definitely worth doing because it's more quantitative. But yeah, I haven't done that. And I don't think the di portion is interfering in any way with the binding. So yeah, these could be seen as preliminary results to kind of launch into more precise studies with radioactive ox-LDL or like you said, cholesterol esters. But I haven't done it. Mm-hmm. It's expensive.
Yeah. The other question has to do with the fact that has to do with the relationship, if there is any, with fatty acid binding protein. Because obviously
people, yeah,
to transport to move fatty acids around.
Yep. And so HEC293 cells don't express fatty acid binding protein. And that's, again, one of the reasons we used it, because adipocytes, of course, do. And so when you study adipocytes with this pH assay, as you've probably seen from Sue's data, you add the fatty acids and the pH drops, but then it kind of rises back up fairly rapidly, and we think that's because of the intracellular metabolism of the fatty acids. So we specifically chose 293 cells to eliminate some of that. But I know there's papers, and I know people have studied the fatty acid binding protein, but nobody's found any interaction yet, specific, just with CD36. But it doesn't mean there's not an interaction. Dr. Atkinson, or?
I have two questions for you, Anthony. In your SPR experiments, you're presumably using the extracellular domain.
That's right. I forgot to mention. Yep.
Which is an important point. So,
yep.
What is the evidence? Maybe some of the inference might come from the LIM2 structure, but what is the evidence that without the two transmembrane domains to cement the C and the C terminus together, that the conformation of CD36 is sustainable?
Yeah, no, that's a great point. I was supposed to mention that, but I forgot. So with my SPR experiments, we're using HEC293 purified human recombinant CD36, but it does not have the transmembrane domains. Again, the intracellular domains are tiny, but yeah, I mean, so I can't really speak to how that might be different.
The LIM2 structure, they are actually in opposition to each other, and Daisy Sharman's data, I think, quite clearly shows that the two transmembrane domains are interacting with each other in SRB1.
Yep, and they're palmitoylated. There's 4 palmitoylations, so
right.
Yeah, and that's another advantage to. I mean, it's an advantage to purifying the full length and then comparing it to the our other SPR data.
So the other question I have to ask you, why does CD36 enhance oxidized LDL uptake, whereas SRB1, which is
Similar.
pretty homologous to it,
Yep.
functions in selective transport of cholesterol ester out of LDL without taking up the LDL particle? Why?
I don't think it's known. Yeah, it's a good question. I certainly don't know. But I don't know if it's known because I have looked into
How
that.
is the uptake done? Is it endocytic?
That's not even known. I have checked into that. Yeah. I don't know. I assume it is, but that doesn't mean it.
Well, some of the questions Haya was asking you if you actually looked at radio-labeled ApoB on LDL.
Right. Yep, and that might help distinguish for sure. Yeah.
Perhaps answer that.
Yeah. Again, that would make it even more important to do those kind of experiments because they are more precise. And again, it's a pretty rough assay. It does prove the point or show the point that it's the binding and it's enhancing the uptake, but it doesn't give you those fine details. Yeah. So, but it does put those in the now. Those are, I think, priorities also. Yeah.
I have other questions, but I'll
Okay.
doesn't matter.
Yes.
I was wondering about whether SPR is the standard technique in the field to distinguish non-physiologic sticking of fat acid versus the physiologic binding of fat acids. I think hot not binding to CD36 is a good indicator. It's specific, but is it the norm in the field?
It's not. So this is a new assay that I developed. So using SPR has not been done, with cyclodextrin solubilized fatty acid. So that's one of the reasons I had so many controls, because this is a new assay for that type of binding. It's a difficult thing to study fatty acid binding on proteins, and especially with all the background binding that fatty acid gives.
So what are the other techniques that other scientists?
Boy, I mean, that's one of the difficulties, right? So with those uptake assays, again, they were using fatty acid that's radioactive, but they were just measuring end products. And that's why we use the fluorescent assays. So if there was a more precise assay, somebody would probably be using it even for that. So I don't know. And there was a question in the back, yeah.
Does CD36 oligomerize on a cell membrane?
Nope. Not that I know of, no. It does cluster, and one of the reasons we wanted to do Q-dot experiments is because it travels along F-actin, and there's a Nature paper showing this from Harvard where they used Q-dots on CD36, and they studied the tracking of CD36, and it definitely is influenced by oxidized LDL, but they didn't look into it much. They just showed that it tracks on F-actin. But people have done cross-linking studies, and there's no direct interaction with, it doesn't oligomerize, like you say.
But do you think it's possible that fatty acid increase the clustering of CD36 and then?
Yep. It's definitely yep. I think we had one more. Do we have any more questions or got time? Yes.
Yeah, so let me follow up on one of the earlier questions. I'm still having trouble visualizing how exactly the binding might potentially occur. I understand you don't know the details. So it seems like you think that the AFABP homology domain is involved in the binding of CD36 to oxidized LDL.
Yeah,
But
correct.
this is a barrel, so one would expect that this barrel might potentially pick up a couple of fatty acids and then sequester them, and then how does the rest of the binding occur?
It's a mystery.
Does
Yeah.
an LDL fall off, or perhaps there is another domain on CD36 that might be involved in LDL binding? I mean, is there any relationship whatsoever between LDL binding to the receptor and the FABT homology in your field?
Yeah, I mean, it's a great question. That's why we have a biophysics department. I don't know, but I think it's a good project. I mean, that kind of detail. I don't even know if I can speculate.
Yeah, I thought there was a basic apex in the structure of lymph.
A basic
Kind of apex structure created by several helices at the end opposite to the transmembrane helices of the lymph protein, which was proposed by Cassini research throughout the family long with
Yeah.
receptors and H receptor and several others. So I would have thought that perhaps the apex domain, which is highly helical,
Right.
and not the
Yeah.
would potentially be the binding site. But
I know if you can answer that, you can answer Dr. Atkinson's questions too. I mean,
no,
it's kind of the same.
You're an expert.
this-
I wonder what you think.
I can only speculate. Like I say, this is crude. It does show the binding. Good. All right, thank you. Unless we had one more.
Add to the questions.
No.
The talk. Well, I didn't interrupt and add anything. The talk spurred a lot of very good questions, and the grants that I was writing while he's writing his thesis addressed some of the questions. We're looking at cells like pancreatic beta cells and macrophages that normally express low levels of CD36. We're inducing higher levels, and we're measuring cholesterol esters chemically. And it's known in pancreatic beta cells that cholesterol can accumulate to the extent that it crystallizes. So these are excellent questions. You know, what actually happens and what's the pathological effects. These are model cells to address questions that nobody ever bothered to address, and we're moving into more physiological things. Thank you for your attention.
Thanks.
Thank you.